Hello, everyone. A warm welcome to our audience to the 27th webinar of the GFI Ideas webinar series, and this is more SciTech focused. Before we begin, I'm Dr. Akshay Bhatt, Science and Technology Specialist working with the Good Food Institute India. And with me, I have Dr. Padma, who is also a Science and Technology Specialist, and she's an expert in the plant-based protein side of things. So before we begin the webinar, you as a participant can use the Q&A button for questions you will have for the speaker during this presentation. And also feel free to use the chat section for any comments you want to make. Please do not use the chat section for any personal discussions or off topics. And also after the webinar, we'll have 10 minutes of Q&A session where all questions which all the participants will have, they can go live and ask those questions. There is a raise hand button which you can press, and we will allow you to ask any questions or clarifications you want to have. And for those participants who are not yet familiar with the kind of work our organization does, the Good Food Institute India is a part of a global network of nonprofits with 6 key regions. Basically, we are based out of 6 key regions: India, APAC, Israel, US, Brazil, as well as Europe. And as an organization, we work with entrepreneurs, scientists, investors, and other stakeholders, diversifying protein sources for a more secure, sustainable, and a just food system. So we do this across 3 pillars: business, science, and policy. Very briefly, under business, we have innovation as well as corporate engagement. In innovation, we help early stage entrepreneurs as well as startups by helping them in terms of, let's say, all the way from ideation to launch, in terms of fundraising, in terms of product development, hiring, and so on and so forth. And also in terms of corporate engagement, we consult with some of the world's largest food companies to help them align and venture into the alternative or the smart protein space. Under science, our SciTech team has expert scientists working to advance the open source research in the space by setting up, let's say, coursework, by giving out grant calls. For example, the annual grant call which we have, which is slated to come out in early 2023. And also we work with institutions, organizations, research organizations to build centers of excellence within the university ecosystems. And under policy, our policy team advocates for fair regulatory pathways and also works with the government in terms of unlocking funding for research and development in this space. So a very brief introduction about Dr. Bhavesh Kumar Jani was joined us. Dr. Bhavesh is the head of the Department of Food Process Engineering College of Food Technology at Sardarkrushinagar, Dantiwada, sorry. I hope that I'm pronouncing that correctly. Dantiwada Agricultural University, Banaskantha, Gujarat. So Dr. Jani graduated as an agricultural engineer and completed his master's in processing and food engineering from the Junagadh Agricultural University, Gujarat. And during his early career, his focus was on byproduct utilization as well as underutilized crop product processing. He's keenly working and promoting indigenous crops as a promising food and nutrition source, and currently he's focused on developing peanut protein isolates and hydrolysates with variable functionality suitable for, let's say, preparing, for example, textured flour as well as plant-based beverages and foods. He's also involved in developing low-fat, high-protein peanut butter, for example, utilizing byproducts as an ingredient. He's also produced 2 successful entrepreneurial ventures with his technical expertise, and he has several publications as well as book chapters under his belt, and sits on the editorial board of several, let's say, top national and international food processing journals. So a warm welcome to you, Dr. Bhavesh Jani, and it's over to you now, Dr. Bhavesh. So I'll stop sharing my screen and please go ahead with your presentation. Looking forward to some exciting developments from your end.
Thank you so much. Is it visible now?
Yes, Dr. Jani, it's visible.
Presentation is visible, right? Thank you. And good evening, everyone. One and all present here. Let me come to topic. Especially, it is divided into 3 phases. One is the indigenous sources for plant protein. Another is the scaling up of it, and by improving the functionality of the protein, scaling up the same at commercial level. So my purpose and where I am serving as Akshay has already introduced very well, so nice to him. In addition to that, we are promoting indigenous sources because of the demand of the industries itself, and not for us. Another thing is our farmers. So it is a clear thought that how an industry and a university or research institute can collaborate and work out on something which I mean closely related to the price parity that you can see here in this slide. You know the how far today we are not at par with even conventional animal-based sources, and that's why we have to pay a larger amount not only for the product but also while procuring the raw material or ingredients. So it's an industry demand. From their side, we came to know that we need to develop some proteins, particularly plant-based proteins, which should be at par with in case of cost, in case of ingredients, and as well as in case of products at par with the available conventional animal-based products. So friends, when it comes to indigenous kind of thing, we'll discuss later how the indigenous and what are the products or what are the agricultural commodities which can consider. For now, let us see. The thing is, plant-based protein should be a sustainable. Like sustainability depends on the taste and texture, of course, it is, and as well as the price or the cost of the ingredients, right? So far in my study and the industries with whom I am connected, particularly plant-based product processing industries, they are, I mean, saying that the ingredient prices are rising day by day and the indigenous ingredients are not being promoted. So that sustainability is the main issue to promote the plant-based as well as alternative protein sector in India and abroad. So our total focus for today, I confined it to India and somewhat part of Asian countries where we can I mean promote our indigenous crops and the ingredients being processed from them. Moving forward, as we know very well that this plant-based, and not only plant-based, but there are other alternatives as well available, which are, I mean, like now in use in case of alternative protein sector and product processing. So in my view, I divided it into 6 parts, particularly plant-based, microbial or single cell or insect-based proteins, as well as the cultivated one in laboratories, cell-based, of course, and the composite one. Composite in my this criteria, it is the composites of plant-based as of now. So that whole will make alternative protein ingredient as a promising sector, right? So in case of plant-based, just we can see typical issue. If I compare when it comes to plant-based, people will always tell like nutrients, yeah, of course. So that ingredients lacking in the essential amino acids and so many things are there. Yeah, you can see in the chart from the bottom to top in a single column and in all the columns, it's a comparison from the plant-based raw ingredient, raw material, not the flour or concentrates or isolate. It's a simple comparison of the seeds and the existing animal-based options. So friends, you can see how this essential amino acid, I'm not going to take each of them, but the small amounts and the marginal increase in the amounts as compared to other the animal-based protein sources that can be seen clearly, and this you must be knowing about it, right? So let's come to a closer picture, and the closer picture says the plant-based seeds, particularly agricultural seeds, which are the promising plant-based ingredients for protein processing and protein extraction, particularly to prepare protein ingredients. Here also I highlighted a few selected plant proteins, and of which few are of my study as well, so I considered it here. So friends, the pea and soy, the soybean itself, I took this as a seed. So where the opportunity lies, particularly friends, you can see the opportunity lies in processing side and not the raw material side. Raw material itself is having almost equal proportion of each of the amino acid. Of course, the PDCA score will vary. That also we'll see in the next slide. Okay, before that, PDCA score, just have some observation and views on the essential amino acids, particularly which are to be said as plant-based ingredients are poor in it, right? So our purpose in our research and our further commercialization of plant-based ingredient is to improve and make it largely available to in our products as well as our body. Now, friends, when we come to plant-based scenario, the products particularly, so plant-based meat and dairy are the promising two sectors where it is largely grown in case of product side. Other than these are also dairy alternatives, particularly cheese and logs and eggs, egg and logs. Everything is available in market, but that cost or that's having a share of a few $1,000 of course, $1 billion. But major share is by plant-based meat and plant-based dairy side. And whatever the ingredients we are focusing today itself, it would be, I mean, definitely a short word that I'm going to present now, but that itself includes application in both the dairy and meat and log, and accordingly we are optimizing it. Now it comes to indigenous sources. So it's almost our first question should be like, what are the indigenous sources? It's something that in around to us, and it is growing in the place of origin to us, right? And that is why it is so required. Why we can focus on indigenous sources. Since years, it is being sown without any kind of pesticide treatment or any side of chemical fertilizers, and so it can be grown in the area. So one can say it's a natural. Another thing in case of natural is a non-GMO side, right? So we can grow our seed itself, and we can grow, we can stock it, we can grow it, and we can sow it by ourselves only. We are not to be dependent on any seed producers or so. That's what the natural thing is in it. Another thing is eco-friendly. So yeah, of course, as I told you, the pesticide demand will be, pesticide requirement can be reduced, and chemical fertilizers can also be controlled. Now, of course, there are tremendous, questions are there in this statement, of course, but it is up to us that since 40-50 years we are, adding chemicals every time in the soil, and through the soil it is into water. So we cannot directly come to something in natural way, but by time it can be done. So it's an eco-friendly option. And it's sustainable in the sense of right from procuring the seeds to sowing to a whole agriculture practices that can be controlled. Something is migrated from somewhere and is being grown, the climate cannot help, and the bacteria, the soil bacteria and particularly soil profile cannot help to grow, and that's why you need to add additional fertilizers and so. So focusing on indigenous sources, that is again not only natural or eco-friendly, but also sustainable source. It can grow easily, and we are habituated with that. These days we face some for our local, particularly plant sources, we face some issues like allergens and allergenicity. But friends, it is a fact that if we use our own ingredient and our indigenous sources in our food and practices, definitely it will not make any kind of health issues, particularly to the people in the surround. And that is one of the objectives. That's why India is considered as a pioneer in case of International Millet Year 2023. It is declared in their inaugural session as well, because we grow the millets in abundant amount, and since ages of years we are growing it, right? So we and our climate are habituated with it. But somewhere out of Asia there may be possibilities of allergenicity or other issues, right? So all these three can be considered together how to adapt indigenous sources. Which are the indigenous sources that India do have? So it's I considered everything within it, but sorghum, millets, rice, particularly chickpea, PGNP, barley, that I found through the sources and through my observation that they are grown since years and years from within our country in our farms. Other than this, pseudo-native crops. So again, it is not possible that we can come directly to the millet and every time we can have that much of abundant production that rather than utilizing in our food, we can go for protein extraction. It's also not an economical way to divert to alternative protein from those sources which are not abundantly grown these days. So which are the other sources? It is the similar kind of sugar native. I narrated it intentionally because these are the crops which are grown and sown in our country since years, but may not be from our native side. I bifurcated in 6 ways. Wheat and rice are the staple cereal crops which are being consumed, and due to already it is high in price. That's why their protein concentrates or isolates processing industries are not so far grown in India. Other than that, soybean and peanut I considered both in a similar category, being an oil seed and category. Also, soybean is also promising, and these days, worldwide tremendously being utilized as an alternative protein source. But peanut also have that possibility that we'll see later. In case of chickpea and mung bean, it's pulses which are in use and in highlight these days. So many times I've came across that why don't I do work on development or commercialization of these sources? But only issue is the raw ingredient cost. Chickpea and mung bean, even it's cost too much in consuming as such in dal form or any other practices. We cannot directly move to that of other like protein and protein concentration isolates kind of practices. In case of maize and potato, it is having little amount of protein, even though it's huge in production. So it could be a little byproduct, and some companies in India and abroad are also working on it, and they're producing protein itself. But again, the issue is the sustainability. And next is the amaranth and finger millet. These two I kept in millet categories, and it belongs to, of course, but these two ingredients are of importance when we consider like how to promote millets, right? And why amaranth? Because I've seen in practically as well as through the sources that amaranth is a promising protein ingredient, and not in India, but in abroad and everywhere it is being studied. But so far, the commercialization of protein processing and protein extraction is not possible, and that may be due to the research and the collaboration from the industries and academia. So that is again the lacuna, and that will cover. I've already started to work on it, and definitely will come to some result by 6 months or a year. And hemp and cotton trees are also, these are having digestibility far good, more than even 90%. But again, the gossypols and other toxic substances present within it, the conversion of that protein to edible, and that would be again a part of research, and that can be a successful thing in future. Now it comes to whenever we talk about plant proteins, of course we need to focus on the digestibility and PDCAS. You know very well the digestibility. So from the data available and from a few of analysis that we have, I've produced some protein values. You can see here that egg and other this animal sources protein, and as compared to that our plant protein in seeds, how much it is present, and not only digestibility but PDCAS is of importance when it comes to a science base, right? So we have seen that digestibility is not an issue. You can see the red columns. It is far better and up to equal to in case of animal based proteins, as well as within plant based there are a number of you can see the commodities. So it is almost equivalent to it. But when it comes to PDCAS, so some like peanut and other millets are deficient in some of the essential amino acids, and that's what directly affect to the PDCAS value. But for that purpose, as I just kept in my first slide, the composite protein, particularly composite plant proteins, you can admixture 2 or 3 proteins to make it to a certain acceptable level. So that is also a possible thing. Now, sustainability lies, again, as I told, it's an issue. When it comes to sustainability, there are challenges that plant-based industries are focusing to work on, and through those people and through those review, I came to some conclusion that I'm showing to you. The first most challenge in sustainability is the taste. So the taste that can be overcome by three-step process. The product that we want to highlight or develop, suppose it is a meat analog, or it's a beverages or dairy alternatives, cheese alternative, egg alternatives. So decide the products, cover up it with the flavor and textural characteristics, so that it can taste like it was an actual a mimic of those available in the market, and optimize the process, optimize it for particular composition and processing purpose. So composition should be optimized first, and then processing can also be optimized. So that is what our work is. Another thing that I want to add, that flavors are available in abundant. The natural extracts is going to be available these days, and so many, producers, small scale producers are also connecting for the same, for the natural flavor as well as the yeast based extracts. So that composition and the processing parameter again should be optimized for a better taste. That is the challenge one that can be overcome. Another challenge that is the more I'm focused for the industry, it is the feasibility. So when it comes to plant protein, yes, the sources are there. Abundant agriculture production is there. India is in the first or the first three rank in the world for almost all kind of plant sources, and that's why as a researcher I'm focusing on plant-based things, right? And so it should be an economical. The first thing is the raw material cost. As we've seen in the earlier slides, that mung bean and chickpea and other sources, the final protein will cost you around 700 to 1000 rupees per kg. Who is supposed to consider it as sustainable? It cannot be. And the final product cost would go beyond the limit that you can imagine. So again, the raw material cost is a far of important. Another is the processing cost. One like potato or maize protein that may be 4, 5%, 3% or 2% protein available within it. You cannot go for sustainable views in case of this maize and potato protein, and it's also a challenge, particularly when it comes to sustainable production. And the cost benefit ratio, that whole thing will be considered, if we can say raw material cost, processing cost, and all the cost, and ultimately the product that you are supposed to sell. So industry has that focus that it should be an economical option. It should be a viable option when it comes to an economy. Another thing is the technical aspects. So available technology is not a sufficient solution because of that poor performing plant-based protein ingredients these days. The research gaps, particularly, which are unhighlighted, or if highlighted, they are only in the laboratories and not on the commercial scale. So that's again a big gap, the technical soundness that we require if we are supposed to consider any indigenous sources of plant-based protein. The compatibility of the ingredients, it's not necessary that everything can be, researched, and the output can be taken like protein ingredients, and that cannot be a suitable option every time. So that can also be studied, and that should be focused. Again, it comes to market. So the demand or the potential, how once it comes to, as the topic itself is for scalable option. So when it comes to scalability, of course, demand or potential of the products, of the ingredients should be considered. If the industry don't need, you need not to go for any process. And next is the adaptability. Adaptability is also an issue, like this, particularly policy issues are there which can hinder the adaptability of the indigenous sourced plant-based protein. The last one is the operational. That is operational, that I can say it's an individual industry's issue, and building a proper team with proper technical knowledge who can continuously be active in case of research and development and the innovation side. And again, the raw material availability. So that is an issue in case of indigenous sources that we already discussed. The third and the most important challenge is the functionality. I have, kept few of the functional properties here, but that's not too far. The only focused functional properties. But in some sources and through some references, I have collected this information for particular like emulsification or gelling. Almost similar technologies are available for the improvement, and the functionality is the major issue when we consider adoption of plant-based proteins. Right? So let's move ahead. Functionality again, as I told, this is some data that you should, focus on. For kind of applications, there are kind of functionality that we require, and that is available in animal-based options. They are also incorporating plant-based options meat tenderizing and other purposes, the improvement in nutrition. For that purpose, they are already adding plant-based sources, protein or plant-based source carbohydrate or other products, right? But when it comes to application to particularly plant-based protein and the quality requirement, functionality requirement for that, you can see the maximum thing lies to solubility when it comes to liquid, dairy or beverage market, right? Dairy alternatives or beverage market. When it comes to meat analog, there are few of the properties like particularly emulsification, water binding, dough forming, and gelling properties are of important rather than heat stability and texture. So texturability is one of the aspects, particularly industry demand from us to have some improvement and development in that case. So let's move to next. Food properties. These are the sensory, and these are the four aspects where all your plant-based food products lies. And that is, 50% of the product is maybe composed of this plant-based ingredients, and that's why it is important to study the functional terms which are associated with these food properties particularly. So when it comes to functionality, it's not only the functional, but the sensorial characteristics that we should focus. That particularly haziness and typical of flavor, beanie flavor, that should be masked or overcome or by anyhow vacuum kind of processing or somewhere it should be removed anyway. And the hydration characteristics, particularly wettability, water absorbing capacity, oil holding and water holding capacity, these are the important characteristics when it comes to meat analog processing, right? And then again, the next is the surface characteristics like emulsion processing, emulsifying capacity, forming capacity and forming stability, whipping capacity. Everything can be considered in surface aspects. And in case of dairy analogs, particularly egg alternatives, it is for more of focus. When it comes to structural characteristics, it's the elasticity and cohesiveness, chewiness, particularly are of important in case of meat analogs and so. So structural is majority towards the texturability and chewiness of the meat analogs, and the ingredient particularly being used in meat analog, they are of important. So we focused a few of these functional properties to develop and to modify our existing indigenous sources for its better adoption. When it comes to production side, so initially there are 5-stage processes that an industry usually follows, and being a researcher and even students who are joined with us, they should focus on it. So each of these 5 processes are having individual scope of research and together as well. So when it comes to raw material selection, we already discussed that depending on the product, the suitability to that product, you need to select your raw material. And that selection of raw material, once you finalize the after post-harvest processing and preparation of that raw material for particularly that quality and that quantity that you want of that quality, that is of important. Having that, you can now come to extraction process, where there are 3-4 again processes are there: physical, chemical, enzymatic, and combined of physical and enzymatic or chemical and enzymatic processes are available, and or one can go for R&D in that. It is an extraction part where protein is extracted particularly. Now it comes to concentration. I mean, particularly fractionation of the protein and isolation from all the ingredients. That is again by precipitation, by membrane separation or hydrolysis, we can go for it. The most important, again, the term comes is the drying, particularly, which is under focus when it comes to ingredients. Drying definitely affect your final quality of plant protein ingredients, particularly when it comes to solubility, wettability, or water holding capacity. And due to the drying process, sometimes the probiotics plant protein ingredients, that is the new word, but of course it is in, demand. The probiotic or fermented plant protein ingredient cannot be processed, and there is a thing behind that even spray dryer also reduce the total count of that particular probiotic foods. So lyophilization, freeze dryer can be utilized, but again, it comes to freeze dryer. It is a commercial viability is also a question. So fluid bed dryer is a routine dryer which is being utilized in commercial scale in the industries. And finally, the applications, you can study all this behavior to the particular applications that you are focused with in meat analogs, beverages, and other alternatives. So friends, it's a process chart that you must be aware about. Now, after extracting the oil, if it is a oil seed, otherwise go for dry or wet grinding. Dry grinding, that will lead to the fractionation and the removal by dry separation or air separation of those carbohydrates, fibers, whatever molecules, and by screening and sieving, you can concentrate the protein. Now, of course, the protein may not go more than 65-70%. You can consider it as a concentrate. So plant protein concentrate can be produced in dry grinding process. When it comes to wet grinding, you are adding the water, and particularly at peak alkaline condition, you are extracting it, and followed by precipitation, concentration or separation of protein using centrifuge or some mechanism. After having that separation, you may be having other byproducts that you can utilize, and further it will go to protein separation and washing for neutralization particularly, and spray drying. So this is how the isolates are processed, and the other things or other byproducts are processed. And that's where the economy lies, particularly if we consider a commercial scale plant protein extraction mechanism. It will cost you around 30-40 crores. And if you are not focusing with the byproducts in that, neither it will be an environmental friendly operation, nor it will be an economical viable operation, right? So always consider first that where your byproducts will be utilized. Otherwise, the project may go to failure. So again, going to depth, extraction, there's a conventional or non-conventional extraction, how it actually works. So, friends, very well that plant cells are typically having covers over the ingredients, particularly carbohydrates, fats, and protein. And that cover is to be removed. And as more as the cover you remove, you will find that as much as the extraction will be increased. And particularly in commercial scale or scalability comes, extraction or yield is of importance, right? So moving ahead, you can see here, this is the available within the cell block. It is available within, and vocal it is available. So you can break this cover, you can break this cover as well, and you will have like extraction phenomena that you are supposed to work on. Finally, the concentration or you can say separation will be there, drying, and ultimately you will have your protein isolate. So this is what the process is. So ultimately, whether it is alkalination, whether it is enzymatic, whether it is physical treatment, the ultimate goal is to obtain or to break the cover over the protein anyhow. So that is what the extraction mechanism is, and everything lies within it in the industry, plant-based protein processing industry. So in our case, what we particularly did that is of your importance. So defeated peanut flour was first procured and analyzed for its characteristics, and how much fat and protein is already there in it. The trouble that we felt that fat and particularly carbohydrates that we need to remove. And the next is the preparation of flour. We added water in the ratio, it is shown here. At a particular pH, it is extracted in alkaline condition using NaOH, and it is then centrifuged so that the carbohydrates particularly that can be separated, and supernatant which was available was taken again for further precipitation. It is precipitated using HCl at 4.5 pH. It's an isoelectric point for the protein, and the extract which is collected, that pellets, is again neutralized using NaOH and washed with water. Then again it is dried. The two dryers in our study that we utilized, the first one is the freeze dryer, another later at the commercial scale, as it is always, it's a spray drying mechanism which was utilized. And in the study of the solubility and particularly emulsification capacity of the product that we obtained was done. So what we observe in case of our work in particular defeated peanut flour, and why defeated peanut flour, it was also a question for you, other than this allergenicity that is always an issue. Of course, friends, as I told you, I am focused with the countries, Asian countries, where this allergenicity is quite low, and it's not at all an issue. Other lactose intolerant and other people are more than that of the intolerant to that of peanut, right? Whatever we did is economical, first I need to say, because it is a byproduct. Defeated peanut flour, peanut is particularly in India is utilized for two purposes. One is for oil extraction, another is for peanut butter processing. HPS is also there, but majority is exported and being utilized in namkeen industries within India and outside the India. So three purposes I can say. One is the hand-picked and sorted, peanut butter processing, and oil processing. So first two are having the raw ingredient self-utilization. The third one is the defeated peanut flour, which is a byproduct of oil processing industries, is collected, and that will cost you even less than 10 rupees. When it comes to a throughout processing industry, the byproduct defeated peanut flour is almost nil cost can be available. So preparing it and isolating the protein, we faced some situations or some observations that we need to overcome in our first phase. It was a low yield. The yield was around 78 to 80%, and we need to increase to more than 85% at least for our requirement. The solubility was poor in earlier case because it's a simple isoelectric precipitation process, and the foaming characteristics even was poor, and the emulsification capacity was also poor. And the possible reason that we found out in our analysis is the poor extraction because of the separation of the carbohydrates and fats was not good as much as we needed or we require in case of to go for commercial scale. And no pretreatment was given to rupture those cell walls that we already seen in the figure. The another thing I found is the acid alkali induced structural stresses. Particularly that alternative acid alkali conditions without any other pre-treatment that disturb the protein structure, and that also caused poor solubility and poor functional properties. And at last, neutralizing it, we could obtain insoluble protein powder. Plant protein powder is having that issue of insolubility when it comes to a neutral pH. So the pH also plays an important role when it comes to its utilization. So another aspect that how the pathways, I can say, how we can improve the particular functionality of this protein. So some references that I found that these are the processes, particularly three processes: physical, chemical, and biological or enzymatic process, we can say. So in case of physical processing, heat, ultrasonication, high pressure processing, pulse electric processing, cold atmospheric plasma processing, and irradiation are in use, but still except heat and ultrasonication, all those four processes are on lab scale or pilot scale. So that is the first observation of mine. Chemical, in case of chemical fibrillation, I found somewhat to be focused. Otherwise, other processes I am not acquainted with, and that's why I omitted it from my research. The biological or enzymatic kind of where the possibility lies. So I went for alkalis particularly because I found at the end the carbohydrates were in measure, and that's why the protein availability was reduced in case of yield. So in the current work, I added enzymatic treatment along with ultrasonication. This is also a pictorial view how this chemical and physical techniques can be bifurcated. And in this heat also microwave, that is one of the possible method by which heating can be done, but that again denaturation or coagulation of protein is an issue, so it should be considered before. So what I did, I added after preparing the particular floor for extraction, before extraction I went for ultrasonication treatment followed by enzyme addition, alkylase addition, and that gives us yield, increase in yield as well as improvement in particular functional characteristics. So the same process I followed after that, and the rest of the thing which is remaining, and we research or we work with collaborating with the industry. So the next part is we handed over to industries and we are waiting for their observations and suggestions. After in particularly solubility analysis, we handed over to them and at different pH values, because it was a requirement from them at which pH value particularly it is better soluble and within the considerable limit in case of commercialization. So that will be done, and at last we'll go for in vitro digestibility and PDCAS evaluation, which is a requirement of a scientific database. So somewhat photographs I want to show to you. The protein extracted in alkaline condition, it is the soluble protein from defeated peanut flour, and the protein concentration is around 78 to 82% in that phase. And finally, it is centrifuged and dried in freeze dryer in case of filler trials or the initial trials on the lab scale. This was the observation. The next one is the particularly commercial scale. So when we were focusing and we were getting trained with laboratory scale, I moved to industry, and at industry scale level, I produced one batch for just to have a study on it. And in that case, whatever the mechanism I utilized in that industry nearby to my area and collaborating with them, and we finally produced bulk of powder for next phase applications for the industries. And in response to that, industries also gave some good highlights, rather I can say, particularly dairy analogs. They've prepared alternative cheese they prepared for using our product. It was amazing and it was praised. So we are also waiting for a collaboration and commercial multiplication of our products. Also, for a new or innovative product side, in our college we are working on plant proteins by developing omega-3 rich plant proteins. It is the first of its kind, and that is also 10% soluble, and it is a particular work on existing plant proteins, because it's an industrial requirement that existing pea or soy protein isolate, can it be utilized for such innovative products or not. So we have that facilities that we utilized and developed some omega-3 rich plant proteins. The source was flaxseed oil. Another plant protein source, particularly millet, that we have focused this year only, that is the amaranth flour, and it was ground flour from amaranth seeds itself. The similar process we need to follow, just you need to change is the protein extraction, that is the alkali extraction point that we found 9 pH is better in our filler trials, followed by all those centrifugation, precipitation, and another centrifugation process, neutralization and spray drying. And our purpose is I found that amaranth has the promising texturability protein in it, and particularly when I tried to a single screw extruder at another place, I found that it can be textured, and for that purpose I went for this particular extraction of protein from amaranth flour, and we have that success review maybe 6 months or a year it will take for finalization of the product in the market. So what we'll do is the optimizing all these parameters. We can go for texturizing using greens production available with us, and texture floor will be prepared from it. Then also in the reviews, it was found that the extrusion also like is a heat treatment, pressured heat treatment, which can produce a soluble protein in it, and the emulsification characteristics can also be improved. So we'll study for it, and further in vitro digestibility and PDCAS evaluation will be done. So that is our target or traits: texturability, water holding capacity, and gelation. That will be focused particularly in case of amaranth protein concentrates that we are preparing. Now, when it comes to scale up, I think almost things are covered, but you just see, this is a big name. Kerry Group has one analysis, and they, published it. So what I observed here, the majority of focus is not in line with the PDCAS, allergen, or texture, or plant-based even. It is lying with the taste, the quality of the protein, the naturality in the ingredient, and that's where our millets or our indigenous sources can be promising, right? And the flavor of the product that can be masked, it's not at all an issue these days. So these whatever the above 40% is, I can say significantly above average, and then that is to be focused, rather than we're focusing in our research in our laboratories on those PDCAS and so on, right? And the PDCAS and others, these scores can be improved by, as I told you, incorporating, and also it is a fact when it comes to a scale up or commercial applications, they are using number of ingredients. So it's not like that our protein is having everything and then only it can be used, right? So that is to mine. I have taken a lot of time, Akshay. Hello.
Yeah, sorry, I was on mute, I believe. Thank you so much, Dr. Bhavesh. I think a very, very informative webinar, especially for people. I obviously think that most of the participants are from plant-based background. I don't have that background. So I think this was a really very informative presentation personally for me, as well as when this goes out on YouTube. I think a lot of researchers, entrepreneurs will definitely find this helpful. I don't see any questions in the Q&A box, but I will invite our participants to ask any questions if they want to ask them live. So Dr. Azim, Ravi Narvani, Swastika, do you guys have any questions for Dr. Bhavesh?
There is one question in Q&A.
Yes, please go ahead. Yeah. Why hasn't consumption and or processing of millets pick up, despite India being the largest producer?
True, Ravi. The only issue is the promotion. Not only millets, but our indigenous crops. This is a particular seminar when we, I and Akshay were discussing on the things that our ingredients are not being promoted, and that's why not only ingredients, but the industries are not being promoted. So it's a promotion. The promotion is the drawback behind this question of millets to be picked up. Another thing is the industrialization of novelty or innovation. So when it comes to millets, we are only focused with something which is to be used like cereals. It has many more than that. It is a nutritious crop. So nutrition and isolation of those nutrition is to be focused for particular consumption. And by this year 2023, of course, the millets will be picked up like anything. If you want to go for it, you just go for it.
And thank you, Dr. Bhavesh. So yeah, year 2023 is the year of millets, and hopefully we'll have some very exciting news in terms of research and development, which we cannot reveal right now, but probably early next year we'll have some good news for researchers in the millet sector. through GFI India. And we also have a second question from Swastika. I mean, she's basically asking, she doesn't have a technical background and whom she can get in touch with, but I'll allow her to speak as well. So, Swastika, you can talk now.
Hi, I'm Swastika Deshpande. I'm based out of Mumbai. So actually, I have great interest in this subject. I also attended the GFI summit that was held. But problem is that I'm not from the technical background. Hence, I'm facing problem in, understanding the product and also kind of getting the pilot product ready. So anyone I can get in touch with or any university who's providing help for me to develop my product.
Are you an entrepreneur or a student?
I am an aspiring entrepreneur, so I want to develop the product, and I am right now in the ideation stage. And I'm also getting incubated from IIM Bangalore.
That's great. I mean, already this incubation and such facilities are available in our university. Also, Rural Business Incubation Center is available for the entrepreneurs like you. Other than this, nearby to you, there are a number of universities which are offering this incubation centers and incubative facilities. You can opt for that, and particularly when it comes to plant-based or alternative protein sectors, it is still under development phase, and I think Akshay could respond to you more in that.
Great. So I'd love to get in touch with someone who has the technical knowledge and guide me on this subject.
You are more than welcome in that case. GFI is the tremendous platform, and not only that, even I am available for any kind of help you want, because we promote entrepreneurs, and you must have seen the presentation. I focused on how a layman to an industrialist can understand, and not particular as a researcher.
Sure,
So,
sir.
yes.
I'll get in touch with you over LinkedIn. Is that okay?
Yeah, it is all right. LinkedIn is the best platform one can engage.
Great, sir. Thank you so much for the presentation. I could learn a few things, and it was really good. Thank you so much.
Somewhere, Akshay, I also want to add, these are the collaborative works, and some information and data particularly cannot be disclosed. But if anyone personally is interested to grow more in that case, I'm ready to share to them and personally to help them as well.
Sure, Dr. Bhavesh. So, I mean, we are happy to make that connect anytime, right? So, obviously, in terms of confidential information, I think it's between both parties. For example, you and the entrepreneur. So the entrepreneur. So we completely understand that. So we will definitely channel any such requests to you and other plant-based researchers wherever there's compatibility. Okay. I have a question for you, Dr. Bhavesh. So, I mean, see, what I feel is that in terms of the plant-based, let's say, especially milk formulations, one of the biggest challenges which many startups face is basically the aftertaste, right? So they try to mask it with, let's say, higher sugar content. For example, I've tasted a couple of products with probably one with sugar, one without sugar. Both taste really very different, right? So the one with sugar, I liked it because obviously it had a really good taste, because probably the aftertaste was masked by, for example, sugar in it. But the one without sugar, it was like, yeah, I mean, I'm not comfortable with that kind of thing, right? So, I mean, are there any specialized methodologies, for example, to mitigate such aftertaste problems? And also, the follow-up question for you is, the follow-up is that, for example, you're working intensively in terms of pea protein, for example, right? So, is that protein neutral tasting, and how did you get to that if it is neutral tasting?
Sorry, aflatoxin. I think it is in the question box. Yes. How to tackle with aflatoxin? So, as I told, the selection of ingredients. Aflatoxin is a mycotoxin which is there since the beginning. It's not like the aflatoxin comes to at a defatted peanut flour stage or at extraction stage. So it is there. Within India as well as abroad, there are countries which are having aflatoxin marks. The residue limit of extra aflatoxin is already defined, and is being in touch with the industries. Now the aflatoxin issue is not at all a major issue. It was there before 5 years, 10 years. Now the moisture content of the particularly groundnut, is kept below 7.2%. So aflatoxin will not be an issue. It's an issue when the moisture exceeds to 8%, and there are chances of development of those mycotoxin within it. So as of now, aflatoxin is not an issue. And even though if it is an issue, as I told, the selection of raw material ingredients itself is a technique to go for it. Now, and again, Akshay, it was your concern, yeah. So in that case, microencapsulation technology, you may be aware about. As in my one of the slides, I presented the same. Not only flavor or aftertaste, beanie taste or bitter taste, but masking the flavor. This can be good options, but when it comes to heat treatment or pasteurization and so processing, everything goes minimized, and at last you will feel that flavor, right? So microencapsulating those flavoring substances, microencapsulating those omegas or those vitamins can be a promising option to overcome the aftertaste and nutrition factors after processing. And so that is one of the options. Another is the novel technologies that we can go for it, least heated or non-thermal processing technologies, because covering the substances or masking with the flavors is not the solution. That flavor should be there even after packaging and during selling. So people are adding those flavors and making something better, but the processing itself is too poor that those flavors cannot be retained. And if it comes to at the end, microencapsulation can give you a promise that slow release of those flavors, and that particularly will cover up this problem.
Yeah, thank you so much, Dr. Bhavesh, for that answer. Yeah, probably we'll surface this when some entrepreneurs ask this question. I'm not sure whether they're actually pursuing that, but yeah. Yeah, Dr. Padma probably can comment more on this. I'm not an expert in terms of microencapsulation of plant-based proteins, but I think Padma also has a few questions.
Yes.
Please go ahead, Padma.
Hello, Dr. Bhavesh. Excellent presentation. Thank you for that. So I have a couple of questions, and I completely agree with you on the encapsulation as an approach for masking the off flavors. So the only thing, I think the choice of suitable carrier materials to do that would help in that. True.
It's a practical thing I observed in my products, which I developed from plant-based ingredients.
Yes. So I have two questions. Like, you had presented a process flow diagram for the production of plant protein isolate. So what do you consider is the bottleneck process or a critical control point in that entire process?
Particularly pH. That is the first during alkalination and during coagulation or precipitation. I need to add a few things for other people as well. When we come to commercial or scalability things, this isoelectric precipitation is the technology as of now which can be commercial, which is already commercialized, and mechanisms are available. When it comes to advanced or novel processing technologies like PEF and so, as well as other extraction like fermentation or enzyme-based processing, it still need research and pilot scale studies to be proven studies, sustainable outputs. So when it comes to critical points in total research in isoelectric precipitation method itself, pH is the first thing that one need to have in mind. The another thing is the drying or drying temperatures and the exposure to those dried air. That is definitely an affecting factor to the final products and particular functionalities of those proteins.
Yes, exactly. That was my second question that I had written. Like, what have you compared, like spray drying and freeze drying? So what is the influence of each of these techniques on the protein functionality?
Yeah, so initially in case of freeze drying, the first factor was again scalability. another factor is it yields, the quality, particularly visually, it was like, something best. But when it comes to solubility, it was at par with the spray dried product. The second is the processing time. Nobody is going to, invest their 7-8 hours to up to 18 hours for those, tons of commodity or tons of material. So I faced that that even freeze drying, if even promising and yields, better product in somewhat functionality, I can say emulsification particularly, and water holding capacity, it was good as compared to spray dry. But when in toto we can consider, when it comes to statistically proven thing, then it comes to spray drying, which can be commercially adaptable.
Yes, that's true. Freeze drying, the batch processing, as well as the vacuum
For laboratory
deployment.
analysis, yeah, of course, optimization of ingredients and the beautiful functional properties, you can go for freeze drying, but that can only be possible at lab scale.
Thank you, Doctor.
Thank you, madam.
Thank you. Thank you, Padma. Thank you, Dr. Bhavesh. And audience, one last chance. If you have any questions, you can raise your hands. Otherwise, we can close this webinar. Just giving like 10 seconds here.
I think it is a recording and it will be there on YouTube. So
Yes.
there will be definitely questions afterwards, and some strong discussion and commercial feasibility and so. So I'm sharing in chat box, I'm sharing my email ID where you can put any questions or any query that you want. It's an official ID, as well as my LinkedIn account name.
Yeah, very helpful.
So
Thank you so
anyone
much.
can connect to me. Then, yeah, as I told you, I cannot share everything on the screen. So I personally can help him or her out wherever it is necessary.
Yeah, very helpful. Thank you so much, Dr. Bhavesh. And since we don't have any additional questions from participants, we at SciTech GFI India would like to thank you heartfully, Dr. Bhavesh, for making time for this presentation, and I should say that deeply enlightening in terms of the information which you have shared through your presentation with our participants. I'm very sure that every participant really enjoyed this presentation. And to all the participants, we have already sent the link to our SciTech community. Please fill in that form. It's not going to take you more than 5 minutes, 3 minutes, but please do put in your name and your designation, where you come from, what's your background, so that you don't miss out on any, let's say, updates in terms of what's happening in science and technology, not only in India but also globally, in terms of the smart protein or the alternative protein sector. So thank you so much, Dr. Bhavesh, once again for joining. And we'll be ending this presentation.
I want to add something. I've gone through your LinkedIn profile, as I was not aware much about you, and I found interesting that you are working in the field or in the side as a scientist, which I haven't explored till that. So that would be great combination. I'll learn from you as well. And I'm thankful to GFI community, GFI India community, for this chance, and that will be highlight our indigenous sources and promising sources from our country itself. Thank you, Dr. Akshay, and thank you, Dr. Padma, as well, for being here. Thank you all the participants.
Thank you so much. I thank you so much. I'm going to stop the recording now.